Hello friends, in this video lecture we will discuss about steps of plant breeding technology. As we earlier discussed, plant breeding is a purposeful manipulation of plant species to get a new cultivar or new varieties that have a number of traits or characteristics, like there may be presence of disease resistance, there may be better or more productivity, there may be quality productivity. Such type of characters must be there. So what steps we have to follow for obtaining such type of plant by plant breeding method? So there are 5 steps that all are given here. First, collection of variability, or also known as germplasm collection. Second is evaluation and selection of parents. Third, cross hybridization among the selected parents. Fourth, selection and testing of superior recombinants. And fifth is about testing, release and commercialization of new cultivars. Question may be about the arrangement of these 5 sequences. So randomly there may be 4 sequences and you have to arrange them accordingly, which one comes first and which one in the last. Now we discuss one by one about each step. So first is about collection of variability or germplasm collection. So in NCERT the first line that is given is very important. This is genetic variability is the root of any breeding program. Means more genetic variety or diversity that we have will help for improvement or to achieve new cultivars or new varieties of crops. That's why this is the root of any breeding program. Means there may be plant breeding, animal breeding. So for any breeding there is first need of genetic variability. Second, many crops have pre-existing genetic variability from wild relatives of the crop. So as we discussed, all our major food crops are derived from the domesticated varieties that still we are using today. So these domesticated varieties also have some pre-existing genetic variability from their wild relatives of particular crop. Third is about collection and preservation of all the different wild varieties, species and relatives of the cultivated species is the primary need, is a prerequisite. It means this is the first thing that we have to do first in the direction to achieve a new cultivar and for maximum use or effective use of naturally available genes in a particular population. The entire collection of tissues, seed, plants having all the diverse alleles for all genes in a given crop is called germplasm. So what is germplasm? This is sum total of all genetic material that is present in a particular plant. And this can be obtained from seed bank, gene bank, nurseries where plants are growing, etc. So collection of variability or germplasm collection is the primary thing, is the key thing or is the root of any breeding program. We have to collect all the varieties of an individual species from all over the India and if possible then also from other parts of the world. Second step is evaluation and selection of parents. So after collecting germplasm, we have to evaluate and identify plants with the desirable combination of characters. The characters what is needed for further or future growth. The plants having desirable characters are called as parents that we have to use for the next coming generation. During evaluation and selection of parents, we can also create pure lines whenever desirable and possibly needed. So what is pure lines? These are progenies of a single homozygous self-pollinated plants, or we can say pure lines are a group of closely related individuals of identical genetic combination. After completion of these two steps, the next one is cross hybridization among selected parents. What is hybridization? Hybridization is simply fertilization where there is fusion of two opposite type of gametes that are coming from their respective parents, male and female. Before hybridization, there is gametogenesis that is followed by meiosis most of the time, and meiosis is known for crossing over and recombination that causes genetic variation, and these variations are responsible for evolution or here for advancement of new varieties. So hybridization is the most common method to create genetic variation, desired characters of parents combined by hybridization. For example, suppose there are two selected parent plant varieties A and B. Parent A is carrying high quantity of protein while plant B is carrying disease resistance. Here in plant A disease resistance is absent, while here in plant B their protein quality is very poor. So after mixing these two or after hybridizing these two, there will be a plant that will carry high protein quality and disease resistance. So this is offspring C that carrying characters from both the parents, and here there is a vigor, there is a advancement with respect to their parents. Same here, there is another example. This is plant A1, this is plant B1. A1 is carrying quality grains while B1 is carrying quantity grains. Here in A1 quantity is very poor but quality is very high. Here in B1 quantity is very high but quality is very poor. So individually they both are not beneficial, but somehow if we add characteristics of both these plants in a single plant, that will be highly meaningful to us. So during hybridization there will be generation of a new plant that is C1, and this carries characteristics of plant A1 and B1, that is high quality and quantity of grains. Like same, there may be mixing with other characters, like there is a plant that is dwarf in nature, there is a plant with tall nature. When we are trying to fuse both characteristics in a single plant, so as we simply know, most of the time small crop plants or semi dwarf varieties are very important for us because they take very less time to grow or reach at their maturity. So flowering, fruiting will be earlier and there will be more production with respect to consumption of natural resources. Hybridization is very time consuming and tedious process because first of all we have to germinate seeds, then there will be growth of that particular seeds. So as we already know, in plant life cycle there are three phases: juvenile phase, reproductive phase and senescent phase. So juvenile phase is considered as pre-reproductive phase and senescent is considered as post-reproductive. So first plant have to achieve their reproductive phase, and this phase is variable for plant to plant. All these phases will be shorter for those plants which have a smaller or shorter lifespan, but this exceed for those, or this is high for those plants which have longer lifespan. For example, suppose there is a plant that lifespan is for 4 months. So here all the phases like juvenile phase, reproductive phase and senescent phase will be for shorter time duration. But suppose at the same time there is a tree that have lifespan of 50 years. So here all these phases will take huge time for completion of that one. And in flowering plants, before flowering there is no sexual reproduction. So at the time of sexual reproduction we can implement all these experiments. So that's why hybridization is a very tough, because for hybridization flowering is needed, and when there is flowering then we have to take desired pollen grains collected from male parent and we have to transfer this again over the desired stigma of female parent. This may take huge time to reach at flowering condition as we earlier discussed. So this is first point, means we have to wait till flowering in a particular plant. Second thing, that is again very time consuming. It is not necessary that the hybrids do combine the desirable character. Only one in few hundreds to thousand crosses. So the desirable combination, means when we perform for 100 to 1000 times to achieving a particular character in a hybrid form, then out of 100 or 1000 there may be rarely one chance when these characters are getting fused with each and come together in a new cultivar. For example, a wheat variety that we discuss, HUW 468, this took 12 years to develop. So we can imagine about the time needed for hybridization or needed for improvement of a new cultivar. So cross hybridization may involve a single cross, that is in between two parents, or multiple cross, that is in between more than two parent plant. This all is because of need or demand how many characters we can successfully transfer in a single plant. That will be a new cultivar. So now about types of hybridization. As we discussed, hybridization is simply fusion of male and female gamete. This may be categorized here into these four. First is intravarietal. So intra means inside. So inside the varieties. So this occurs in between two plants of the same variety. Means in same variety there may be two plants where hybridization we have to perform. And this intravarietal hybridization helps in improvement of self-pollinated crops. Second is intervarietal, that also known as intra-specific. This occurs in between two varieties of same species. So here species is same, that's why intra-species. Here variety is same, that's why intravarietal. But this intra-specific is in between two varieties of the same species, that's why intervarietal. And this intervarietal helps in improvement of both self as well as cross-pollinated crops. The third is interspecific. This is in between two different species from the same genera, that's why intrageneric in nature. For example, Saccharum barberi and Saccharum officinarum. Saccharum is genus, so in between the same genus, that's why intrageneric, and barberi as well as officinarum, these are two different species, that's why this is interspecific. So this example given in NCERT, so that's why very important. Now the fourth is intergeneric. This occurs in between two different genera. So two different genera, and genera is under family in taxonomic hierarchy. So in between two genera of the same family. This type of hybridization is rare and practiced to create new crop species. For example, when there is hybridization in between brinjal and tomato, there is formation of new cultivar, that is bromato. Second, potato and tomato, there may be new cultivar pomato. Same, there may be other intergeneric varieties. So when we are performing hybridization, this involves a number of steps. First, selection of desired parents. So we have to take parents with a specific type of character that will help to improve the characteristics of new cultivar. Second is selfing. So when we pick a particular plant, there may be some very important characteristics, there may be some very desirable characteristics, and these characteristics may be segregated or separated at the time of gametogenesis, at the time of cross fertilization. So to continue to achieve the same characteristics in next generation, we have to perform self hybridization or selfing. This will cause homozygosity of that desired characters. We will perform the same thing till obtain the maximum quality of that respective character. Third is emasculation. This is about removal of anthers from a bisexual flower before maturation, and this will help in prevention of self-pollination. As we know, bisexual flowers carrying both sexes, and when they both are getting matured, there may be selfing or self-pollination in between. To stop this, why we have to stop this? Because we have to transfer desired pollen grain on desired characteristics of stigma. So self pollination is not needed at that time. We are actually discussing about artificial pollination, where the desired pollen is getting transferred over desired stigma. The fourth step is bagging. So after emasculation, after removal of anther from a bisexual flower, we have to cover that flower with a proper bag which is porous in nature, that allow only gaseous exchange, not pollen grains, and this will also prevent for unwanted pollination. Otherwise, if emasculation is done, pollen grains of undesired plants or undesired flowers may also reach over there. So to stop this, we have to perform bagging. Next is tagging. So we have to tag properly by writing every and each step that is done till that with proper date and timing. Without tagging, because there are so many plants in that particular area where plant breeders are performing the same thing. So for proper identification, tagging is must. Otherwise, they cannot identify that the particular plant is performed up to which level of plant breeding. And finally, the last is artificial pollination, that is crossing. Now we have to transfer desired pollen grains on matured desired stigma of the emasculated flower. So by this there will be pollination that will successfully that in future may successfully proceeds up to fertilization, and then we have seed. That seed is further cultivated and next step of plant breeding will continue. So fourth step of plant breeding is selection and testing of superior recombinant. After completion of third step, that is cross hybridization in between selected plants, there will be a number of seeds, and from that seeds we have to select the desired superior hybrid by careful scientific evaluation of the respective progeny. Maximum time there may be more than one superior progeny, and that selected plant, the selected progeny or the selected plant is again self-pollinated for several generations till they reach a state of uniformity or homozygosity. This also known as to achieve pure line. Again, pure line is a progeny of single homozygous self-pollinated plant. By continuous selfing, there will be accumulation of a single type of characteristics or accumulation of same type of characteristics generation by generation. This is known as pure line. So selection is the oldest method of crop improvement. This point is very important, means there may be direct question, which is the oldest method of crop improvement. So this is selection. Now the fifth and final step of plant breeding, this is testing, release and commercialization of new cultivars. So newly selected pure lines that we already discussed here are further evaluated for their yield and other agronomic characteristics, like there is presence of disease resistance, there is presence of pest resistance, there is presence of drought resistance, the plant is carrying maximum productivity, the plant is carrying quality productivity. We have to check, or plant breeders have to check all these characteristics. This testing is done by growing these pure lines in research field in ideal conditions of agriculture. So what is ideal conditions of agriculture? It means we have to irrigate properly that crops, we have to provide proper nutrients, proper manure, proper temperature. Everything must be according to the need. And the same testing is followed further in farmers field for at least 3 growing season at several agroclimatic zones. What are agroclimatic zones? So these are specified area with specific environmental condition. So we have to test that particular cultivar in all those areas where plant is growing. This new cultivar is evaluated in comparison to the best available local crop cultivar in that particular area. If this is superseding over that cultivar, it means the variety, the new cultivar what plant breeders developed is best type, and this is ready for release and commercialization in open market.